University of Pavia, and the title of his, the evoking title of his talk is Multiscale Brain Modeling.
So thank you very much for the opportunity to present this part of our work. I will concentrate actually on brain modeling carried out using computational tools. And I will face in particular the issue of multiscale, which means the issue of complexity that derives from the organization of the brain. To be, let's say, more clear, I would like to present the layout of the talk. I will start with the foundation of the issue. So why we need computational brain models? We have heard today about theoretical models or theoretical interpretations of brain functions, firing activity, and so forth. But computation is an important complement to all this activity. And I think I will be able to show you examples of what computation about the brain can actually do. The multiscale problem from cells to brains, that's another sort of issue that is haunting all the scientists. So we needed to connect what the neurons and the brain are doing, but to explain one, we need the other and vice versa, which brings about problems like the inverse problems or the reconstruction problems that are not easy to tackle. Then we have to consider what a neurobiologist normally does. So he poses questions that are not exactly the questions of a computationalist. We need to understand in neurobiology when a neuron is activated, what a neuron is doing in terms of functionality, and how the neuron is working. So we have to go at the level of causality on how the neurons generate the computation. That is something that in many cases is not considered in high-level theories, where you skip all the causality factors and you jump directly to the description in terms of statistics. So the story, if you take a biological perspective, is even more complicated. And then I will consider the complementarity of two modeling approaches, the bottom-up, which means starting from fundamental biophysical principles of molecules and neurons and climbing up to networks and brains, or vice versa, observing what the brain is doing as a whole and going down to the level of neurons. In order to make the story easier to understand and to follow, I will exemplify it through examples rather than explaining all the different steps individually. It would take a life. And I will take most of the examples from models that we're doing on the cerebellum because we're working on it. But the cerebellum is a prototype of brain networks, and then we use these cerebellar networks into the brain models. So you will see all the brain and not only the cerebellum in the end. Final, but last but not least, how these multiscale models are useful for biotechnology, for ICT, for medicine, with future developments to be discussed and open for questions. Before starting, I would make a sort of anticipation on the theories of the brain, because we have seen theories on how neurons make their discharge or the spikes are correlated in time and space in the brain. Actually, the brain merits theories, and these theories run around since more than 1 century. Most of these theories have had a short life, because they immediately confronted with new biological discoveries that demolished the theories or dismounted the theories. But there is something that is interesting and that I would like to show you, because it could be also connected to the computational models. The brain can be conceived as a complex, adaptive, predictive system. That's the very basic. This means that the brain generates internal schemes that are transformed into actions or behaviors or faults. And then the consequences of these schemes is to generate a feedback. For example, if I take this bottle, I predict the weight, I predict all the sensory consequences of my action, and the actual sensory consequences fed back to the brain. What does the brain do? It compares my prediction with the execution, with the actual state of the system. And in doing this prediction, learns from what? From the interaction. This learning through interaction or through trials and errors is typical of certain brain structures. And these structures include the cerebellum as a typical structure that is working at this level. And therefore, the cerebellum will come back into my presentation also because it is the critical element to bind the prediction with the consequences of this transformation of prediction into action. This idea that was born more or less 2 decades ago, and then was formalized more recently, has got a final set out through this work that I would like to mention. I will not comment too much. It is the theory of the free energy principle minimization through Bayesian active inference, which means that the elements in the brain or the components of the brain are able to generate a representation of states, the sensory state, the internal state, the action state, and these states cooperate one with each other to get to the control of the hidden state in the environment. Once this happens, we have the feedback that I mentioned you in the previous slide. This is a formalization in which this free energy, that is not the free energy of thermodynamics exactly, it's very similar, but an informatic energy. This free energy is the parameter that is controlled by the brain. The brain tends to minimize free energy, which means minimizing the surprise. Surprise is a component of entropy. So basically minimizing free energy, which means minimizing surprise, is what the brain is actively achieving. All the brains, if they have a project in nature, have the project to minimize free energy. And by minimizing surprise, they improve the success of an action, and they reduce the deleterious effect of an error. This is why we learn.
Sorry. Yeah, that's the question. Why you call it free energy?
It's called free energy for questions that are related to informatics and is loosely related to the concept of free energy in the second law of thermodynamics.
Yeah.
It's the energy that you can use to carry out a work on the external world, basically.
It contains a pure, let's say, an energetic part and then an entropic one, taking into account of the solid. That's why.
Exactly. So don't try to find exactly the correspondence with free energy in thermodynamics. Is a computational definition. Okay, so recently we applied this principle to the cerebellum connected to the cortex. We applied it into models where we have adopted the free energy principle and Bayesian active inference at the level of single neurons, making plasticity the synapses, and that works. It is able to predict actually the occurrence of subsequent states in the interaction between the brain and the objects. We have applied it to some simple behaviors, and the principle works. So now the paper is submitted. We are looking at the consequences of this idea, but basically this is a theory based on a principle, and we've done a computational test to see whether it applies to a real circuit. So that's what happens. Good. Now go directly on the level of what biology is telling us or what we expect from biology about the understanding of the nervous system. We have to answer these questions: when circuits are activated, what they do and how they work. And go directly to an example. This is the multiscale organization of a circuit of the cerebellum at the level of molecules, microstructures like a synapse, macrocircuits made of neurons connected together, mesoscale circuits including different components of the cerebellum and also extracerebellar structures, large-scale structures, which means the cerebellum connected to the whole brain. Clearly, there is a reverberation of effect from the low level to the high level and vice versa. So we have to understand how this reverberation happens and how the different levels interact one with each other. Clearly, what we measure at the different levels is very different. We see single neurons when we stay at the neuronal level. We see ensemble signals like the EEG or LFP signals when we go into the tissue, or when we stay outside of the tissue with an MRI scanner, we see fields of neurons all working together, but not exactly the neurons, indirect measurements of neuronal activity. So we have different levels of precision in getting to the understanding of activities in the brain. About the operations that happen in the circuits, they're quite complicated, and through biological and physiological investigation, one can define them. It's a bit painstaking, but there are tens of papers trying to understand what the neurons do in the circuit. And actually, we have generated terms that you all know, like synaptic plasticity, when the synapse changes the weight of transmission, or action like pattern recognition, where we have neurons working like perceptrons. And we have phenomena of binding between neurons that generate coherence, oscillations that generate waves in brain activity. We have phenomena of adaptive filtering when the signals are transformed in the frequency domain, or center surround when they are transformed in the spatial domain. All these things can be analyzed, described, and defined in biological terms. So now we expect to find them back into the models. Because if we lose these things that are critical to allow the functioning of a circuit, we lose the understanding of the circuit. So we are accumulating problems now rather than solving problems. We are depicting a situation in which what we want to understand is very complicated. So you will see the solution that is proposed. At the clinical level, the situation is much easier because the books teach us that when one has, for example, the cerebellum out of order, we do not move correctly the arm or the body or whatever part of our motor system. Now, the situation is becoming more complicated because the cerebellum is strongly connected to associative areas. It has been defined a new kind of syndrome that is called ataxia of thought, that is parallel to the ataxia of movement. And this ataxia of thought means that the cerebellum is performing the same fundamental operation on all the parts of the brain. It's not designed to control movement. It's designed to perform an operation. And this operation can be reverberated at different levels on the motor system, the cognitive system, recently also the emotional system. So it is reverberated everywhere. It's a sort of primitive of computation that the brain implements through circuits, and that it is reverberated wherever the brain needs it. Evolution has reconnected the cerebellum in many ways in different animals, performing different kinds of operations. Okay.
That's the question. What is the what is called the ataxia of thought?
Ataxia of thought means uncoordination of the elements, for example, in a sentence. Instead of putting them in a proper sequence, they don't go in the proper sequence. Or one is not able to coordinate the elements of a complex thought, and that eventually reduces the mental faculties of a subject. And normally when the cerebellum is out of order, both things come out. The clinician normally discovers immediately the disorder in movement because it is macroscopic. But careful analysis at a psychophysical level can allow to discover also the problems in cognitive manipulation of objects, symbols, and whatever. Okay, so this is an example of how one can explore the brain, and in the specific case, the connectivity between the cerebellum and the cerebral cortex. And this was done in our institutes in Pavia, and shows that 80% of the connections from the cerebellum passing through the superior cerebellar peduncle go to the associative cortices, not the motor cortices. It means that humans are not designed to use the cerebellum only to move, but also to think. And of these connections, most go to the prefrontal cortex, the top level, I would say, of our cognitive system. Okay. This is an example in which the cerebellum is strongly activated through in a test of action observation, in addition to be activated in action execution. It is even more activated in action observation than action execution. The subject is doing nothing in the scanner except of looking at another projected on the screen that is doing an action. And the cerebellum activates much more than when it actually does the movement itself. So that's an example. And another case is here when you see the resting state networks in fMRI, in which the cerebellum is strongly co-activated in the default mode network, introspection, the ventral attention network, switching attention from internal to external reference state, and Salles network, attention directed toward an external element of attention. So the cerebellum is not only activated in network performing motor or sensory motor control, it's activated in a much more broader way in different kinds of operations of the brain. Okay. So now we've seen what we can understand and learn from biology, and now we have to try to make models out of this. So the idea is that these models have first to face the problem of complexity, the issue of the multiscale organization. They should be able to answer the when, what, and how issue of neurobiology, and what kind of models can we do? First of all, these models have to be able to bind mechanisms to function and behavior, to be causative in a way. So they have to be mechanistic. In the second point, they have to be able to fill gaps in knowledge through constructive rules. We do not know everything in biology, we know only something. But if we have rules, we can also fill the gaps. That's what normally a model does. It's not telling you exactly what the real world is. It's an interpretation of the world. Finally, importantly, it should be able to generate theories or to test theories in order to verify or falsify the theories. So it's a net, is a sort of tool that we can use in addition to the direct observation of phenomena. Okay. The idea that has been developed in this project that you see here, the Human Brain Project, of which some of us are also members, is that we can generate a reconstruction of the brain from bottom to up. So we can start from the knowledge of neurons, build models of neurons based on the biophysical rules that generate neuronal functions that we know pretty well, then make many of these neurons, put them together in a network, run the network that has the same level of granularity of single neurons. It's only bigger and computational intensive, but it's the same level of granularity of the molecular-based neuron. Then the problem becomes, if you want to grow up in scale, it becomes a bit tricky because we do not have all the networks of the brain represented in this way. And then the computation will be dramatically heavy. So we need simplification. And this is shown here. We can climb up to this level of single neurons, microcircuit models, very detailed. I will show you some example. Then we simplify them and we go to the mesoscale and macroscale. So if the simplification is done in the proper way, we still have a good matching with the biological properties of the brain. Okay, but we're still not in the brain. We're still dealing with networks. Now, the point is, what do we do with the brain? The brain normally starts from MRI measurements, EEG measurements, MEG measurements, and then we have a nice representation of ensemble brain functions or brain dynamics, but we don't understand anything about the neurons. So the point is here, instead of solving the awkward inverse problem that I think is more or less unsolvable in this way, because we should be able to understand what the neurons do from an MRI signal, too many steps. We get lost along the way. We will never get to the right way, the right point. So the point is that we can inject these spiking models into virtual brains constructed on the base of a connectomic system. So we generate a connectome. In the nodes of the connectome, we put some neural mass or mean field model, but then we can inject, in place of these neural masses or mean field models that are abstract representations of neuronal activity, we can inject or insert components that are spiking. This is a hybrid model. And in this way, we will have neurons that are like the real neurons, or networks that are like the real networks inserted into virtual brain models. You will see in a while how we do it. In parallel, we can develop controllers for robots in which all the control system is designed as in robotics with equations and functions in hardware or software. But then in some parts of the system, we introduce spiking networks. That's another way to have the neurons running inside a sort of artificial system. What comes out of it is an understanding of brain functions in a way that we can then discuss what it is. And then there are applications to biomedicine, ICT. Okay.
Sorry, just to understand better, these spiking neurons that you put on the on a connectome in practice are macro neurons anyway.
Micro.
So are
I show you now. Are micro neurons. You basically need
so how many, just to understand?
Oh, you can go up to tens of thousands.
Okay.
Yeah, tens of thousands, which is still small scale compared to the brain, but then, okay.
But I have another question, sorry. But the controller that you have on the bottom is just for understanding, is not mimicking a brain function.
Well, the controller is designed, in the case of robotics, is designed to fulfill some operation. For example, you wanted to do a motor controller, no? And you constructed the motor controller to have several elements like the motor planner, the inverse model of motion and of the plant. And then you have the cerebellum. The cerebellum, you can make spiking. Then you have several nuclei for the feedback. There are Kalman filters to generate state estimation and so forth. So you have the normal controller of a robot in which you place a spiking element. Okay. This is an example of the workflow. And you see here a neuron that is reconstructed based on elementary properties of the structure of the neuron, of the membrane of the neuron, of the ionic channels, the membrane receptors, the synapses that are completely taken from biology. So here on the back of this figure, there are tens of years of work in many laboratories. It's not simple to do, but with appropriate techniques, it can be done. And now we have these neurons for all the cerebellar types of neurons. You see the neuron that is blinking, is making spikes, is working. And the validation, constructive validity and functional validity of the neuron is mostly 100%. It reproduces all the properties that are known until now about these neurons through constructive principles, okay? Then it can be simplified into a smaller version, can be placed into a network through an internal connectivity derived from anatomy. And then what we get is something like this, is a network in which all the neurons can work together, depending on their connectivity and their internal dynamics, okay? This structure can go up to millions of neurons, and we are at the moment going toward the generation of a full cerebellum of the mouse, which is a 65 millions of neurons. So in 1 year or so, we should have the full cerebellum of a mouse. These kind of structures can be embedded into brain simulators, like in the example here, that all these elements I will show now one by one, or in robotic controllers. Each level is paralleled by equivalent experiments designed either to construct components of the model or to validate the model. Nothing that goes to the next step remains non-validated. Everything is validated before jumping a step, okay? Okay. Now I
just validated. You mean validated against random models, null models?
Against biology, against measurements.
Okay.
Against measurements.
Okay.
Yeah. This is the same example. Okay. This is the principle of the mathematics that stays behind the biophysical models and is the basis of all the construction of the bottom-up models. Is differential equation mathematics, in which the membrane is represented by the Ohm's equation, the Ohm's law, written in a proper way to take into account the properties of the neuronal membrane. That's something rather normal in physiology. The ionic channels are represented either using equations of this kind, which describe the gating properties of the ionic channels, or through Markov chain models, stochastic models that represent the change of state of the molecule in time. So there are different kinds of representations that always lead to a system of ordinary differential equations. Same level of representation is given for the synapse, always first order differential equations coupled in a way to represent the vesicle cycle. And then the system gives out a membrane potential in time. That's actually what the neuron is doing. This membrane potential this time is the spike, is the equivalent of the spike activity that is recorded from all the experiments that we've seen also this morning in some presentations. Okay. This is again the same as before. I skip it. Is also only bigger. This is an example on how the validation construction process goes on. This is the neuron that has been sold out as the simplest neuron of the brain. It is not at all simple. The beginning of the story is at the end of the 90s with the definition of the main ionic channels that set up the neuronal activity. We did a model with 7 ionic channels, but one of the states of function of the neuron was not captured. Since the description of the ionic channels was very precise, the conclusion was one channel is missing, not that the channels are wrong. Then we looked for one channel missing that was predicted by the model. We found it, and we got all the functional states known at that time. Okay. Then the story went on because the amplitude of the action potential was not correct. It was smaller, it was bigger than expected. So we had an hypothesis. The ionic channels were not located as we thought at the beginning, were displaced into different parts of this multi-compartmental system. We analyzed the displacement using various techniques. And then finally, we decided to displace the ionic channels in the model. And the model gave back exactly the amplitude of the spike. That was another second model. Then these spikes had to travel along the axon as they should do. They didn't. They didn't properly. That was a bit surprising to us, but they didn't. And then again, other sets of hypotheses and experiments. We found that the ionic channels have a spatial modulation in the axon. And that's this paper here that came out a couple of years ago. And actually, the channels of sodium channels in the axon are different from those in the axonal initial segment. Seems a small detail, but it completely changed the life of a neuron. Without this small detail, the neurons do not work and the brain dies. So that was the other story. And then finally, we discovered heterogeneity in the discharge property of the neurons that is generated by another ionic channel predicted by the model. So but the model is continuously predictive properties. Is really generating a sort of model-driven science rather than a sort of intuition or, let's say, fantasy-driven science. That's quite important. Otherwise, we would have looked for an infinite number of details rather than pointing toward those that explain the real problem. And we discovered another channel. So that's another paper that came out last year. So basically, the story is quite interesting. These models are capable of predicting the functional state of the neurons. And you immediately understand their applicability to pathology. Because if a part of the neuron is not working properly, immediately the model tells you the neuron is not doing what it should. Putting neurons together is another issue. We have developed a simulator that is called the Brain Scaffold Builder, is going to be released now, that allows to place neurons depending on their morphology, identifying by high-level microscopical determinations, and to place them, connect them, and simulate them. This is an example, again, can predict the model once constructed, can predict the activity of a network in space and time, predicting oscillations, coherence, resonance, and in space, the spatial organization of neuronal activity. This is a high-level validation. And eventually predicts the filtering properties of the network. These are power spectra of the signals that come in and out of the network. Something that cannot be, at the moment, cannot be measured. So that's a pure prediction. We'll wait. We'll see. This is another example. Can predict activity recorded in vivo. This is an example in which the model, the spiking network, is placed here inside the robotic controller, performing a multiple set of tasks, and always can generate the answer, a response that is consistent with biology, showing, for example, learning at certain synapses and not at other. Finally, the models is placed into virtual brains reconstructed starting from atlases. For example, this is the famous Allen Brain Atlas. And the model can work inside the real connectivity of a whole brain. That's mouse. So this spiking activity is not recorded in the neurons of an animal, it's recorded in the neurons of a model, of the rat brain. This is the brain of humans in which we need, this is the last figure, the last technical figure, in which we use MRI to generate the structural connectome, to generate the functional connectome. And then when we place dynamic nodes into the virtual brain model, we generate a simulated functional connectivity. Functional connectivity can be compared to the empirical functional connectivity here through an optimization process. And what we get out is a parametrization of the connection weights between the different parts of the brain. And this is, I think, is the most important element in terms of applicability to humans, because this can be done on single subjects. So this is exactly personalized medicine because we can do it on single subjects. What are the improvements to be done? Well, the nodes can be improved. The connectomics can be improved. Connectomics is a story like that was explained by Federico this morning. It can be improved in many ways. And also the functional MRI can be made better or more meaningful for us. On our side, we can improve the nodes, changing, for example, the representation of these nodes that is normally a neural mass. Think about the motion equation written in some useful form. We can transform it in a statistical representation of the mass of neurons, which means a mean field. And we're working on the mean fields. So we're preparing the mean fields to be changed and transplanted into the virtual brain. Well, I skip this. I jump to the conclusion. Now these networks are running into the brain simulators of the mouse, of the human. They are running in cerebellar robotic controllers. They've been transformed into GPU versions, so they can run mostly in real time in the different configurations. And they've been placed into neuromorphic computers in which the architecture is the architecture of the network. It's not the architecture of the Turing machine. So in these neuromorphic computers, we have the neurons that are connected as processors of the computer. And these are all tests that we're doing to transform the knowledge of deriving from biology, passing through computational models and going into different kinds of applications. What's the future? The future is to create a hub of laboratories and places where this kind of operations are performed as a service for Europe. That's the idea of eBrains. That's the platform of the Human Brain Project. And there will be different levels of service, like the supercomputing service, the modeling service, like the one that we're setting up, and data processing services, brain atlasing services, in order to generate a framework for the study of the brain. I stop here. I thank all the collaborators in my laboratory and in the laboratories that are connected to us. And thank you for your attention.
Thank you, Egidio. There is time just for a short question, if there's some, please.
May I tell one thing, or?
Oh, there is someone from the internet.
If I have the way, or I don't know.
Sorry, I stopped my presentation. I do stop sharing and I enable screen sharing again. Okay.
Please, Franca.
Okay, if nobody is speaking, I can just comment 1 second, or do you listen me? Do you hear me?
Yeah, we listen you.
If I can, or there's somebody, I mean.
Oh, go on with your question, please.
No, I was only commenting somehow that the proposal of trying to model the effects of neuromodulation that Egidio commented before is fantastic. Now that I know who you are, because I'm so sorry that I was not aware of your great work, and the idea, if you really think that can be of interest to move really in a model, in a modeling that can show even that these patterns that we are convinced are transmitting information does really exist would be fantastic.
Thank you. I mean, I think we should really get in touch after this meeting and try to understand better what we can do. Yeah.
I'm really fascinated. I will definitely contact you. Thank you so much.
Thank you. Thank you for the comment.
Okay, please, the question.
All right, that's a very small question. It's really fascinating. At the beginning, you showed something that resembles reinforcement learning, which is like you have an environment, you have an actor that based on the observation, there is a policy transforming the observation into an action and so forth. So I want to ask you about, did you arrive to something that can be applied now and maybe compared to the standard reinforcement learning algorithms? See, I see that you are planning to put this stuff into robots or something.
Yeah, basically what we have put in robots is a circuit that performs error-based learning that can be compared to reinforcement learning. Basically, the circuit receives an error signal that's more than an error, it's a teaching signal that tells the circuit that there is an error, there is a mismatch. And these signals allows the circuit, put the circuits in the learning mode. And this is based on neuronal functions, not on our writing of equations that allow the system to do it. These are the neurons that do it. It's a sort of implicit computation of the circuit that clearly we can track back to the level where they happen. No, we also know what happens now. So basically there are synapses where this happens and the system begins to change the synaptic weights. And the system works in a way that optimizes the weights in order to reduce to the minimum the learning through compensatory actions. Just to make an example, I impress the weight of 2 kilograms to this bottle. It's not 2 kilograms, there are 200 grams. And I lift the bottle to the ceiling. So it's wrong. So the error is you're putting too much weight in your movement, no? And the synapses will begin to go down and down and down, reducing the strength until I match the weight of the bottle. Based on what? Based on the recognition of the weight by eyes, through the eyes. So I make a prediction of what I should do. And then after that, my prediction is put into action. If I make a mistake because my prediction is wrong, because I don't know the content, I don't know the fluid, I don't know the mechanics, I don't know anything, then the system begins to calibrate the internal parameters, and then implicitly I learn the mechanics. So basically what is written inside the brain, and in this case in the circuits of the cerebellum that we are using, is the mechanics of the interaction body environment.
Okay, let's